Hello, and welcome to my presentation, Modeling Muscular Dystrophy in Drosophila, a study of lamins and interaction partners. Our lab is interested in the human gene LMNA, which, when mutated, can cause a wide variety of diseases known as laminopathies. These include muscular dystrophies, lipodystrophy, heart disease, progeria, and other rare diseases. Specifically, we are interested in Emery-Dreifuss muscular dystrophy, which causes joint contractures, muscle weakening and wasting, as well as cardiac defects. The LMNA gene encodes proteins called lamins, which provide a structural scaffold for the nucleus and help regulate gene expression by binding to chromatin. There are two types of lamins, A-type and B-type, which form separate networks. Lastly, lamins have three distinct domains: the head, rod, and tail, which contains an IG-like fold. We will be focusing on substitution R249Q, which causes Emery-Dreifuss muscular dystrophy and is located in the rod domain. We are investigating the question of how do different amino acid substitutions within lamins cause such different diseases. We took two approaches: an in silico approach, in which we mapped disease-related amino acid substitutions onto a 3D structure of the IG fold, and an in vivo approach in which we generated a Drosophila model of R249Q in the Drosophila ortholog lamin C, which is the substitution R264Q, and observed for any defects. From our in silico research, we found that disease-related amino acid substitutions occur throughout the entire IG fold. On the left is a cartoon of the IG fold, color-coded by which diseases occur at specific residues. As you can see, every disease located by the different colors can be located in any region of the IG fold. On the right is a surface structure of that same IG fold, complete with the color scheme. This structure also includes predicted binding pockets, which are indicated by these clusters of white dashes. 7 out of 9 of these predicted binding pockets overlap with disease-related amino acid residues, leading us to hypothesize that these amino acid substitutions may be disrupting binding with partner proteins and contributing to the disease phenotype. From our in vivo studies, we saw that the R264Q Drosophila model causes myonuclear lobulations and complete pupil lethality. Looking at lamin C, which is an A-type lamin, we see that the nuclei are heavily lobulated and that the lamins pool into these lobulations, which is quite a stark contrast from the normally tight envelope formed by lamin C. We also looked at B-type lamins, which also exhibit abnormal localization, but not nearly to the degree as these A-type lamins. And we looked at the nuclear pore complex, which exhibits mislocalization patterns similar to that of the B-type lamins. Although R264Q caused complete pupil lethality, we did not observe any significant defects in larval motility. From my studies, we've concluded that disease substitutions of all types map throughout the entire IG fold. These substitutions may alter partner protein binding. We also saw that R264Q causes severe nuclear lobulations, and that it causes both A-type lamins and B-type lamins to mislocalize. Although R264Q did not reduce larval motility, it did cause complete death at the pupil stage. In the future, we plan on expanding these studies to a model of R264W, which in humans causes congenital muscular dystrophy. We will compare it to our R264Q model and try to elucidate whether the difference in these phenotypes of Emery-Dreifuss muscular dystrophy versus congenital muscular dystrophy are due to cytological differences within the nucleus, or perhaps other factors in the genetic background. I'd like to thank my mentor, Dr. Lori Wallrath, as well as Sydney Walker for helping me out so much on this project. I'd also like to thank iCrew and the NIH for funding my research. Thank you.